Artificial Intelligence Doctor. User question. How do different nutrient combinations influence the pulsatile pattern of insulin secretion from pancreatic islets? And what role does this temporal pattern play in maintaining glucose homeostasis? GPT-01 answer. Introduction. Insulin secretion from pancreatic beta cells is inherently pulsatile, characterized by oscillations in both amplitude and frequency. This pulsatile pattern is crucial for effective insulin signaling and glucose homeostasis. Different nutrients and their combinations can significantly influence this temporal pattern of insulin release. Understanding how these nutrients affect insulin pulsatility helps elucidate their roles in metabolic regulation and the pathophysiology of diseases like diabetes mellitus. Influence of nutrient combinations on pulsatile insulin secretion. Glucose as the primary regulator. Mechanism of action. Glucose enters beta cells via glucose transporters, primarily GLUT2 in rodents and GLUT1 in humans, and undergoes metabolism to generate ATP. Impact on pulsatility. Elevated ATP/ADP ratio leads to the closure of ADP-sensitive K channels, causing cell membrane depolarization. This triggers the opening of voltage-dependent Ca superscript 2 channels, leading to calcium influx and insulin granule exocytosis. Oscillation generation. The metabolic processing of glucose creates oscillations in intracellular ADP levels and calcium concentrations, which drive the pulsatile release of insulin. Amino acids. Mechanism of action. Amino acids like arginine can depolarize beta cell membranes directly, while others like leucine enhance mitochondrial metabolism. Synergistic effects with glucose. When combined with glucose, amino acids amplify insulin secretion by increasing the amplitude of insulin pulses. Impact on pulsatility. They can enhance the first-phase insulin response and modulate the frequency of insulin oscillations. Fatty acids. Mechanism of action. Free fatty acids, FFAs, are metabolized in beta cells to generate lipid signaling molecules that potentiate insulin secretion. Short-term versus long-term effects. Acutely, FFAs enhance glucose-stimulated insulin secretion. Chronically, they may impair beta cell function. Impact on pulsatility. Short-term exposure increases the amplitude of insulin pulses, while long-term exposure can disrupt pulsatile patterns. Incretins and gut hormones. Role of GLP-1 and GIP. These hormones enhance glucose-induced insulin secretion and influence pulsatility by modulating intracellular CAMP levels. Nutrient interaction. The presence of nutrients stimulates incretin release, which in turn affects insulin pulsatility. Combined nutrient effects. Synergistic modulation. Nutrients in combination can have additive or synergistic effects on insulin secretion. Complex meal intake. Mixed meals containing carbohydrates, proteins, and fats produce a more pronounced and physiologically relevant insulin pulsatility compared to single nutrients. Regulatory networks. Nutrient combinations influence various signaling pathways, e.g., mTOR, AMPK, that collectively modulate insulin pulsatility. Role of temporal insulin patterns in glucose homeostasis. Enhanced insulin sensitivity. Pulsatile versus continuous insulin delivery. Pulsatile insulin delivery is more effective in promoting glucose uptake and inhibiting hepatic glucose production than continuous exposure. Receptor dynamics. Pulsatile insulin prevents downregulation and desensitization of insulin receptors on target cells, maintaining insulin sensitivity. Synchronization with metabolic needs. Glucose flux matching. The oscillatory release of insulin aligns with fluctuating blood glucose levels, ensuring timely glucose uptake and utilization. Metabolic efficiency. Temporal patterns optimize the action of insulin with respect to nutrient availability and energy demands. Beta cell health and function. Preventing beta cell exhaustion. Pulsatile secretion reduces the metabolic load on beta cells, preventing dysfunction and apoptosis associated with continuous high insulin output. Intracellular signaling oscillations. Proper pulsatility maintains the rhythmic activity of key intracellular pathways, e.g., calcium signaling, essential for beta cell health. Implications in disease states. Type 2 diabetes mellitus. Disruption in insulin pulsatility is one of the early defects observed, leading to impaired insulin action and glucose intolerance. Metabolic syndrome. Altered nutrient-induced insulin pulsatility contributes to the pathogenesis of insulin resistance and metabolic syndrome. Conclusion: different nutrient combinations modulate the pulsatile pattern of insulin secretion by affecting beta cell metabolism and signaling pathways. The temporal pattern of insulin release plays a pivotal role in maintaining glucose homeostasis by enhancing insulin sensitivity, synchronizing metabolic processes, and preserving beta cell function. Disruptions in this finely tuned system can lead to metabolic disorders, highlighting the importance of dietary composition and meal patterns in metabolic health.